Thank you very much for this invitation. It's a great pleasure to be here, to be able to discuss with such a qualified audience. And well, let me share my screen. The work that I'll be talking to you today is the result of a very recent research that I've been carrying since the beginning of this year. It is not published yet as in a journal, but you already can find this in Autoria as a preprint, which is entitled, it is entitled The Legacy of the Southern American Extinct Megafauna on Plants and Biomes. I'd like to, before starting, I would like to thank my collaborator in this study, Julie Pausas, which greatly helped to make this work look how it looks today. And so that's it. Let's start. Sorry. Okay. Well, when we think about large mammal herbivores and megafauna, which here will be treated as synonyms, we frequently immediately think about African savannas with the rhinos and elephants and giraffes and wild beasts. But many people don't know that thousands of years ago, and especially more than 10,000 of years ago, South America was also the home of many large mammal herbivores. Not only South America, but especially South America was the home of many large mammal herbivores, including some that resemble those that we see today, like the Mastodon platensis and other elephant-like animals, but very different creatures too, like the giant sloths and other types of endemic animals that we had in South America. And while most of these animals went extinct at the end of the Pleistocene, we still have some regions in the world like Africa and Asia to give us a lot of insights about the whole of that megafauna plays on ecosystems. So these studies are showing us that where megafauna is abundant, herbivores have a very important role in regulating ecosystems, both ecosystem dynamics and vegetation structure. So this figure here from Pausas and Bond 2020 summarizes these effects. Megafauna may regulate fire regimes by controlling grass species composition and biomass, so decreasing fire frequency and intensity. Megafauna can also influence the nutrient cycling by driving nutrient redistribution and influence the composition of decomposers. And more importantly, megafauna can regulate plant biomass and vegetation structure by generally reducing tree biomass and also reducing tree growth. So these effects suggest that megafauna have a very important role in organizing and controlling ecosystem dynamics. So in general terms, we may say that megafauna creates their own habitats, which are generally grassy ecosystems with very low tree cover, in which fire regimes and nutrient cycling are broadly controlled by megafauna activity. But we still don't know how much of this activity have influenced the history of South America so far, and how the current vegetation patterns that we observe are influenced by these activities that the now extinct megafauna exerted in the past. In these ecosystems that are created by megafauna, we also see that plants are generally possess many trait adaptations that can be used to decrease the amounts of damage that are caused by large mammal herbivores. In this paper, that was published in this year. Me and Julie Pausa suggest some of the traits that are more likely to be associated with herbivory maintained savannas. So in these ecosystems, we are likely to find plants with high wood density, plants that invest more in height growth in relation to a given stem diameter. We also have plants with spines, with cage-like crowns that prevent large amounts of uptake by large mammal herbivores. But these are only some of the potential mechanisms by which plants may defend themselves against the megafauna. Many studies are showing for a long time that these strategies does not seem to combine randomly and does not seem to be distributed randomly in space. Instead, in African ecosystems, it has been suggested that based on climate, soil, and on plant traits, two broad ecosystem types can be recognized. Nutrient poor, moist ecosystems in which plants rely mostly on leaf level defenses, and arid, nutrient-rich ecosystems in which plants are heavily armed against herbivores and have some typical defenses like spines, like small leaves, and nitrogen-based chemical defenses. So this is a pattern that been long recognized for Africa, and we don't know if similar patterns can be identified in regions that once have megafauna as a potential example of anachronisms found in regions that no longer have diverse megafauna assemblages. So one of the objects of this study is evaluating which such regions can be found also in South America. And finding these regions is relevant because it could also help us to find where in the continent megaherbivores were probably more commonly found. So it could give us insight about the distribution of the different types of ecosystems in the continent, especially those that are preferred by large mammal herbivores, which are generally open grassy ecosystems. So in this study that I'm describing to you, we addressed three main questions. The first question was if the current distribution of plant defenses, the current geographical distribution of plant defenses across the continent could be explained by the past distribution of the now extinct megafauna. The second question was if this occurs in such a way, if the effect of megafauna occurs in similar ways compared to what is observed in Africa, so that we can identify two well-defined open ecosystems, one characterized by arid, nutrient-poor soils and plants defended with spines, nutrient-rich soils, plant defended with spines and small leaves, and another with moisture with plants defended with leaf-level defenses, and a third ecosystem which would be equivalent to afrotropical forests in which plants are expected to be generally sensitive to herbivores because of the high productivity of these ecosystems, allowing plants to escape herbivory through height-escape. So we were expecting to find three different regions, two that were influenced by herbivores and one that was not. And third, we evaluated if there was evidence of currently forest-dominated ecoregion being savanna during the Pleistocene, using both megafauna and plant trait information to address this question. So to address these three questions, we sampled 4 plant functional traits. We compiled data on 4 plant functional traits that are known to be related to mechanisms by which plants defend themselves against large mammal herbivores. So we used Leaf spine as an indicator of leaf level defenses, as many studies have been showing that this trait is correlated with other leaf level traits. Plants that have leaf spines often also have chemical defenses, nitrogen-free chemical defenses. We also used stem and branch spines, compiled data on stem and branch spines, which is an important trait to reduce damage from both pulling and breaking the trunks, but also to reduce uptake heights. It also helps with the cage-like architecture to prevent animals from reaching the leaves. We have leaf size, which also reduces the uptake rates by large mammal herbivores. Smaller leaves drive less uptake. And finally, wood density, which was recently suggested by us in a paper to be also a potential anti-herbivore defense trait, preventing lots of damage caused by large mammal herbivores on the trunks and stems. We obtained all the data we could find for the new tropical region, including mostly Central and South America. And after getting all the data we could, we aggregated this data at the species level. When the trait was quantitative, we used the means. When the trait was qualitative, presence or absence of spines, for example, we used the presence, assuming that a species sometimes may not be recorded to show spines in some places because of trade-offs, but that if any individual of the species is found to have spines, it's probably because the species evolved such a trait, although it may not express in every environment. We assigned this mean trait values or this maximum trait values in the case of binary traits to the occurrences of each species in our data set. We generally included more than 2015 species except for leaf spines, which included only 800 species because we only included data from palm species. And we calculated the ecoregion means for these traits, and then we related it with other metrics that we used as predictors in our models related to megafauna, with extant herbivores, with wildfires, hurricanes, climate, and soil. So the herbivore predictors that we used were based on data from the Phyllocene database, plus some extra search for diet information for the species that occur in the neotropical region. We selected the species, distinct megafauna species, by selecting only species that were extinct during prehistorical times, that had a body mass equal or greater to 50 kilograms, and whose diet consisted mostly 9% at least of plants. We also included data for extant herbivores, and here we included not only those that are presently extinct, but also those that were extinct during historical time, were extinct by modern men. And in this case, we did not use a body mass threshold. We included all mammals. And it's important to mention that here we are only talking about mammals. I did not include other groups. So we did not use a threshold for body mass, but we use a threshold for diet, with only including species whose diet consisted mostly of plants. From these variables, we calculated the megafauna richness or extant herbivores richness, the body mass, mean body mass at the ecoregion scale, and the richness of species according to their diet. So the richness of grazers, browsers, and for megafauna species, also mixed feeders. For extant herbivores, we did not include mixed feeders because the data set we used to classify species into diets only classifiable species into grazer and browser according to their dietary preferences. We also incorporated ecoregion level data on climate from WorldClim 2 and included these three variables: mean annual rainfall, mean annual temperature, and rainfall seasonality. We also included soil data from the soil grids dataset. We used the data with 5 kilometers resolution and included as variables cation exchange capacity, soil sand content, and soil pH. Cation exchange capacity and soil pH increase with soil fertility. We also calculated the number of wildfires per area for each ecoregion, as well as the fire intensity using the MODIS product MCD 14ML. For these calculations, we excluded fires occurring in anthropogenic areas and also consider this when calculating the values per area, excluding the areas under anthropogenic land use. And finally, we also included hurricane data because hurricanes have been shown to influence wood density of, hurricanes have been suggested to be candidate factors driving making plants to invest more in wood density. So we also included this variable. And finally, we also classified the species ecoregion on whether they were insular or not, but we only used this data when predicting the factor that explained the distribution of megafauna, because we found that insularity had an important effect on megafauna, both because of the island biogeography theory predicting that island would be species poor, but also because of the island rule that predicts megafauna species to be smaller in islands. So we also incorporated information on whether ecoregion were predominantly island or continental. So based in the results that we obtained, we excluded islands from our analysis with relating megafauna and plant functional traits. So to answer the first question, if the historical distribution of megafauna explained the current geographical patterns in plant defense traits, we used regression analysis using multiple regressions for continuous traits and GLM with binomial family distribution for the binary traits, and performed a stepwise model selection using IC. And based on the best selected model, we calculated the average variable contribution in terms of R square in our models. To answer the second question, if there was globally distributed anti-herbivores, or if we could identify anti-herbivores that were similar to those proposed for Africa, we used hierarchical clustering on principal component axis in the trait by ecoregion matrix. And then after classifying the ecoregions using inertia, we used Kruskal-Wallis tests followed by post hoc Dunk pairwise tests to assess the differences in climate, soil, and megafauna patterns, as well as fire among the identifier anti-herbivores. And third, to answer the third question, if there were forest-dominated ecoregions that used to be savanna-dominated during the Pleistocene, we classified ecoregions as separating those that were part of anti-herbiome containing well-defended species and that had both and that were both megafauna rich and grazer rich, and we interpreted this as ecoregions that had a higher richness of megafauna and grazer higher than the 75% quintile. And then we compared these results with fossil vegetation data, mostly pollen-related data, but also a few fossil soil data. and compared with the current vegetation patterns of these fossil sites and with the vegetation patterns during the last glacial maximum and approximately 20,000 years ago and mid-Holocene in the approximately 6,000 years ago. So this is our data. Here in the first line, you have the megafauna patterns, and the second line, the trait patterns. So you see that we see that megafauna richness as well as grazer richness was mostly concentrated in subtropical latitudes, but also occurred in areas in the area where we find the largest savanna in South America, which is the Cerrado region, and also parts of southeastern Amazon. Uh, uh, the mega grazer richness was mostly concentrated in subtropical ecoregions characterized by forest-dominated grassland savanna mosaics in the continent, like the Chaco region, the Gran Chaco region, in the southern portion of the Brazilian Atlantic rainforest. And we found that grazer biomass was high in a larger region, generally comprising also regions, more tropical regions in South America with seasonal climates. And here we have the trait patterns in which we see the highest values of food density associated with a very small ecoregion of dry forest here, but we see a general pattern that species on eastern portions of South America, including both dry and moist forests, had a very high wood density, and while regions, Amazon regions close to the Andes, show very low values. We see spinescence, we see that the Chaco region together with the Patagonia steppe showed high number of spinescent species, and also a little bit in the northeastern portion of Brazil. Leaf size followed a latitudinal gradient, but not a perfect one. We see that at the southern tip, we have very low, very small leaves, whereas in moister regions of Amazon, we have very large leaves. And finally, we have the patterns for leaf spines, which are based on palm species, showing that regions associated with high altitudes and in the north of the neotropical region had extremely high proportion of spinescent species, whereas some areas also in South American also presented a high proportion of spinescent species. As for our results, we found that mostly for all traits, megafauna had explained an important variability. So we see that for wood density, this variability was very large. No study have reported such a large variability in wood density being explained by a variable at such large spatial scale. So this is very exciting. A previous study have shown, a recent study published in Nature and Ecology and Evolution have shown that the most important climatic variable predicting wood density only explained 15% of the variability in this trait. So it is a really large amount of variability that we find here explained by wood density compared to previous studies. We have also leaf size, which although megafauna is not the most important variable explaining this trait, it's still explaining a substantial amount. And also a previous study published on Science at the global scale have shown that mean annual precipitation explained about 22% of the variability. Here we find that pH was the most important, soil pH was the most important variable, but in our data, we found pH to be negatively correlated to mean annual rainfall strongly, like with an error of 0.78%, 0.78. So since this previous study did not incorporate soil or megafauna variables, it is difficult to compare. but our results suggest that these previous results may be explained by the association with soil pH. And this association that we found here, it's important to mention that the signs plus or minus here indicate the effect. So here the effect of pH is negative, which is consistent with the idea that species tend to have small leaves in environments, in nutrient-rich environments. So even for this variable that's not related to megafauna or herbivores, there may be some influence of herbivores here because herbivores tend to favor plants with small leaves in nutrient-rich soils. We found that stem spines were mostly explained by megafauna body mass instead of megafauna richness, with larger species favoring species that invest more in spines, a pattern that is somewhat similar to what is found in Africa, although there mixed feeders, mid-body size mixed feeders, social animals have an important effect. Here we did not have data on social behavior or the density of animals per area, so perhaps this is why the body mass appears as the most important variable. And we also have another pattern that is similar to Africa, that is the fact that spinescent is greatly associated with drier climates in areas in which mean annual rainfall is low. And we also have leaf spines, for which we found a small amount of variability explained, but this is probably related with the fact that this trait is very labile. There are studies showing that, for example, cattle introduction can lead to plants to produce more spines, especially palms, which are the group we considered here. So probably this is a more labile trait, so this is why we have a lower amount of variability explaining. So, So regarding our first question, if the distribution of megafauna explain variability in current geographical patterns in plant functional traits, the answer was yes. So regarding the second question, we did find 3 anti-biomes, a pattern that seems to be robust to the exclusion of some traits like leaf spines to the analysis, and these patterns were generally consistent with what was predicted based on Africa. We found we gave names to these anti-biomes according to how according to what traits were most important. So the first one is the small-leaved thorny, the small-leaved thorny anti-biome, because it was characterized by plants that have many spines but very small leaves. The second was the leaf defense dense wood antibiomes, which was characterized by species that invest in leaf spines. Here, a proxy for leaf level defenses, it's not necessarily leaf spine. And this is another important why we did not find a strong effect of megafauna on leaf spines, because there are many ways by which species can protect their leaves against, plant species can protect their leaves against large mammal herbivores. And finally, the BLS anti-biome, which is broadleaf sensitive, meaning that species generally have very large leaves, but very little defenses. When we compare the conditions among these anti-biomes, we see support for our initial hypothesis in that mean annual rainfall tends to be much lower in the SLT anti-biomes, while soil fertility tends to be higher, especially when compared to LDW anti-biome, which is very consistent with what has been demonstrated for Africa. And broadleaf sensitive antibiome tended to be moist, but occur on somewhat intermediate soil conditions. So regarding our second questions, if there were globally distributed antibiomes, our answer was also yes, because the patterns largely matched what we found for what has been described for Africa. And we also found that not only climate and soil differ between this anti-herbivore, but there are clear differences in megafauna patterns between the regions, among the three regions. So we see that for the LDW anti-biome, uh, animal assemblages tend to be characterized by a more rich assemblage with smaller species and with a especially higher number of mixed feeder species compared to the other antibiomes. This, uh, these three differences were the main difference in relation to the SLT, small leaf thorny antibiome, which was not so megafauna rich, but had many large species, many grazer species, many browser species, but not so many mixed feeder species. And then we have the broadleaf sensitive antibiome, in which we see low megafauna richness, also large species, uh, low numbers of grazers, browsers, and mixed feeders. But it was not only richness and body mass that differed among these ecoregions. Many studies are showing that especially the SLT ecoregions also seem to have a very particular species composition composed of many endemic species and many species that only occur or that preferentially occurred in this anterior biome. An example is Macrauchenia patagonica, which is a species that had been shown by modeling studies that would probably occur in the southern tip of South America, broadly coinciding with the region that we identify here as the SLT interbiomes. And even in the detail, if we look in detail, we see that, for example, Mediterranean regions and temperate regions in Chile that had not been classified within this anti-biome also wasn't suitable for this species, weren't suitable for this species. So, and also some high altitude Andean areas here. So the distribution of these species closely matched our scheme for antibiomes in South America. Another species, Milodon darwini, also seem to be preferentially associated with this region. So we see here modeling studies for the distribution of these species in the last glacial maximum, in the last interglacial, and in the Holocene climatic optimum. And we see that during these three periods, these species would tend to be associated with the region classified here as the SLT antibiome. And here we have the locations in which these species have been registered, connected by lines that using the pan biogeography methods. Another genus that was common in this region was the Megatherium, which occurred with more than one species. So here is a representation of this species, the size in relation to a human. Here it is the Megatherium americanum. And here we see the distribution of these species, the occurrences of Megatherium americanum, and here other Megatherium species also pretty much coinciding with the SLT antibiome. Here, two other megafauna species that occurred in this region and that consistently occurring during different periods, and it would also occur during the Holocene if it wasn't extinct. The same can be observed by both species. One is a grazer, the other is a browser. And now I'm showing some of these few species that can be generally associated with the other antibiomes, but clearly these antibiomes were the thing that characterized the other antibiomes were mostly related to richness or diet type, because there wasn't many animals that only occurred in this region. But one example is the genus Glyptotherium, which was generally concentrated in northeastern Brazil. We also have the Hippidion principale, which was a mixed feeder that was also associated especially with the LDW anti biome, and also another species of Epidion that occurred in the other part, disconnected part of this anti biome, which show that our results seems to be consistent in relation to the distribution of megafauna species. And we also have the Eremothelium laurilardi that in this case seem to occur in both antibiomes, in the BLS and in the LDW. But if we look to modeling results, this species is especially associated, especially during the last cluster maximum with the LDW antibiome. And here I added the proboscides because I think that many people are curious to see if if the patterns are similar to what is found for elephants in Africa. Here we see that they were very generalist. This species that is very common in the continent, which is the Notiomastodon platensis, occurred in all three antibiomes. And seem to vary its diet according to the antibiome the biome it occurred. So when it was associated with forest, it was generally more like a browser, whereas when it was associated with open areas, it was mixed feeder or grazer. And here, another species, which is the Cuvieronius iodon, which occurred generally associated with Andean areas across the continent. So here is a representation of how the fauna of what is currently the main area of Brazilian savanna, the Cerrado, used to be. It's actually data from the transition from the Cerrado with other biomes in the region. Here is in the transition of the Cerrado with the Pantanal, which is a floodplain savannas. So we see species like Eremotherium laurilardi, Taxodon platensis, which looks like a small rhino, Glyptotherium, and Homecina paulacotoi. And here in the transition between the Cerrado and the Caatinga dry forest in woodland, we see species like Notiomastodon platensis, Equus neogenus horses, Paleolama major, which is closely related to the current llama species, and also Taxodon platensis and Eremothelium laurilardi. So addressing our third question, we found that all this colored area here used to be a savanna during the Pleistocene, somewhere in the Pleistocene, sometime in the Pleistocene. This amounted almost 10 million square kilometers, which suggests that that the neotropics, the South American continent, consisted mostly of savannas during the Pleistocene, and probably mostly during the last glacial maximum, which is thought to be the driest period. But as we are here, probably climate is not a very important factor explaining these patterns. So we can conclude that South America during the megafauna period was much more similar to Africa, which is dominated by savannas nowadays. And we found that almost 7 million square kilometers are currently forests. So these areas have experienced shifts from savanna to forest, from which almost 4 million square kilometers are moist forests, where the rest is dry forests. We also found that almost 3 million kilometers of savannas found in the region today were still savannas during the past. The only region that our data did not allow it to confirm that this was a savanna in the past, but there are other evidence, is the Llano regions, because the Llano savanna region in Colombia and Venezuela, in which we found fewer megafauna and grazer species than our threshold. But but the fact that there are fossil sites from the last glacial maximum suggesting that these areas were savanna confirm that these areas are also stable savannas. Here in these figures, we have several point dots. Red dots are areas that used to be grassy ecosystem during the last glacial maximum. Black dots are areas that were savannas during both the last glacial maximum and the mid-Holocene. Blue dots are areas that were savanna during the mid-Holocene, and orange dots are areas that are savanna today, are savanna patches found within the Amazon rainforest. So from these dots, only this red dot here, these two red dots here, and this red dot here are in areas that are currently savanna. They are savanna, either savanna patches within the Amazon or savanna patches from the Llano savannas. And this black dot here is also in a savanna area. The rest of the dots, except the orange ones, all come from areas that are currently forest ecosystems, but has evidence of being grassy ecosystem in the past, except for those that are in the sea, but they are nearby a forest-dominated ecoregion. So in general, our fossil data, as well as the location of Amazon savannas today, confirm our predictions that these areas were probably savannas during the Pleistocene. And also, there are data that were not included here, but there are many studies showing that in the Gran Chaco region, we also found numerous savanna patches there, although I did not get the exact coordinates at which the sites occur. And in the Caatinga region of Brazil, the dry forest and woodland region, we also find numerous savanna patches occurring there. So this pattern do not seem to be explained by climate. In this study by Arruda and colleagues published in 2008, they tried to see how the vegetation would look like during the last glacial maximum, mid-Holocene, considering available data on climate for this time, for this period. So here we have the distribution of biomes in Brazil today. In Brazil concentrates most of the region that we identified to have been savanna during the Pleistocene. And here are the patterns simulated using data from the last glacial maximum and mid-Holocene. And this paper also considered soil, but although the soil was fixed in the model. So we see that only a small proportion of the forest areas of South America in the south portion of Amazon and also in the northern parts of Brazilian Amazon, we have a small expansion of Cerrado savannas to these areas that can be explained by climate. But in other places, we only see replacement by a type of forest to another type. For example, here, in which we have semi-deciduous forest being replaced by pine forests from Araucaria forest in South Brazil. But these results support our argument that probably it was the megafauna itself who created this habitat instead of a different climate that occurred during that time. So regarding our third question, if there were forest-dominated ecoregions that used to be consumer-controlled savanna during the Pleistocene, we also found the answer yes. So as conclusions of our study, we concluded that current global vegetation patterns are explained by both ecology and history. So history matters, and not only when we are looking to differences in species compositions between assemblages, but it looks like history also matters when we are looking for variability in plant functional traits at large spatial scales. We also found that large mammal herbivores are key drivers of vegetation structure and function, so it should be considered when evaluating the drivers of plant functional traits. And we found that these impacts can last for millennia. So megafauna species have been extinct from South America for more than 10,000 years ago, and when we can still see evidence of their effects in the past, being one of the most outstanding example of anachronisms that we found on Earth, that we find on Earth. And we also found that megafauna, vegetation, and environment interact in such convergent ways that produce and allows the recognition of global distributed anti-herbivores, considering that the same patterns that have been reported for Africa were observed here. We still know little about the difference in megafauna patterns between these Afrotropical anti-biomes, and we don't know if they match what we found here. So this is an interesting direction for future studies. And finally, we found that South America was probably a savanna-dominated continent during the Pleistocene, thanks to a consumer control by megafauna. So we provide evidence that South America looked much more similar to Africa than it looks today. So this is the general ideas that I had to present to you today. I hope you like it, and I'm open to answer whatever questions you have. Thank you.
Thank you, Vinicius. Thank you. I'll give you a round of applause.
Thank you.
I'll just open up my screen. So feel free to, as we normally do in these seminars, feel free to pop your question in the chat, or just indicate that you want to ask a question on a particular theme. And then I'll invite you to, if you can, unmute and show your video and ask the question directly to Vinicius. If for any reason you can't because of problems, or you just don't want to, just let me know and I can field the question directly to it. Just while we're waiting for a few questions to get typed in, I'll kick off. I was quite struck by an absolutely fascinating talk. I really enjoyed that. I was quite struck by the extent to which you were saying the paleo evidence was indicating savanna in what is now Eastern Amazonian forest. And my understanding of this from a few years ago, was that there was evidence of a slight retreat of forest and maybe a corridor opening up around Santarém. But I didn't think the data really was there to show that much of the Eastern Amazon had converted to savanna. So I was wondering whether, is it that there's new data available that you're bringing to this, or is it a new interpretation of the data that existed? That it doesn't seem to be just a corridor opening up or through Santarém, but it seems to be everywhere east of there, largely turning into savanna.
Do you refer to the data, the fossil data, or to our results, specific results?
Well, it seemed to be maybe you can clarify this as well. I thought it was was it the paleo pollen data that was indicating points that were savanna in the the last Holocene.
From South Amazon, we don't have many fossil data showing that this region was grassy ecosystem. We only have this data on the present savanna patches that occur there. So if you look to the presentation, I can put it here again, so okay. it is easier. So here. So these orange dots here, they are dots that are current savanna patches that we found today in the Amazon, and we don't know if it was a savanna in the past. But so the main evidence we had here are these two dots here, which are not very strong because these areas are nearby the Cerrado region, but we also have this dot here that shows that is also a savanna patch in Amazons, but it also shows it's a fossil site and shows evidence for previous savanna states during the last Glacial maximum. So specifically for the Amazon, it's perhaps the region that we have less data. We have much more data showing that the Atlantic rainforests of Brazil used to be savanna. So we conclude that this region is used to be a savanna based on the fact that it had numerous megafauna species and also grazer species, more than the 75% threshold, and because this area was also contains very highly defended species. But in fact, we do not have much fossil information for this area.
Yeah.
At least not that I'm aware of.
Yeah. Okay. Great. Thank you. That brings me on to my second question, which is you use this phyllocene database of abundance and richness. Could you perhaps explain a little bit more about how that database works, how it estimates biomass and species richness overall?
Yes, of course. Well, this database, it is estimation of the distribution of megafauna based on current species, on extant species. So they infer the presence of a given megafauna species if more than 50% of the species that co-occurred with this species during the Pleistocene are present today. So if more than 50% of the species that used to occur together with these species are present in a location, yeah, I'm talking about extant species, then it is assumed that the extinct species also occurred there. There are certain assumptions that underline this data set, and I think the main one is that megafauna was extincted by human influence. So it's not the lack of suitability of the present conditions that would explain the absence of megafauna, but the hunting caused by early humans arriving in South America.
Okay, thank you. Okay, I'll open up to the questions coming in. Then, so Nico Blutskin, you have a question?
Yeah, thanks a lot, Vinicius, for this very fascinating talk. And I'm not much into this topic, but really, like if you look at, for example, the correlations between several traits such as spine abundance and megafauna presence, of course, you always end up in this difficulty to disentangle what is cause and what is effect. Maybe the megafauna follows the Savannah, and then because of the number of spines and so forth. But if you're able to disentangle what the temporal trends are after extinction of megafauna, or maybe the extinction is even not at the same time, can you see that there's like a trend towards like fewer spines, for example, or looking at other traits after the extinction? And can you, in this way, also disentangle cause and effect a bit more? Is it possible?
I think it's difficult because we don't have temporal data. We use species distribution data that are based on several periods, but mostly recent studies. So perhaps if we had data, if I have used data on pollen information to know species composition of a given region in different periods of time, we could look at this information and see. But I think that while in general we can say that it's difficult to trace the causes and responses when using correlation, I think that in our results, it's really difficult to attribute the patterns to other cause-consequence relationship there, because the variables that were mostly most of the I did not show here, but in the paper showing these results, I also addressed the drivers of the megafauna patterns. So I saw what variables better explained megafauna richness, body mass, and so on. And we see that curiously, the drivers of megafauna patterns are not the same drivers of the traits. So we can, for example, fire and cation exchange capacity were two variables very important in explaining megafauna richness, which are not related to the traits that we evaluated. So I think that we can pretty much be confident that the cause there is megafauna.
And the other plausibility you mentioned, like the African scenarios, is well understood, right? So
yeah, of course. But one thing that I think that we could one way of getting deeper in this history, I think, is looking to phylogeny and saying and see if the time at which the traits diversify coincide with some geological event related to megafauna. So this, like Charles Dominic have done in his 2015 paper, I think it's a way, it's an interest follow up for this study.
Thank you. Yeah.
Thank you. Geraldo, do you have a question?
Hi, Vinicius. Congratulations. My question is about the use of climate data from the past. Don't you use it, climate data from the past or only from the present? Because I think it could be interesting to use climate data simulated to the Pleistocene or to the Middle Holocene. What about you? What do you think about?
Okay. There are previous papers already analyzing the relationship of some of the traits with paleoclimate. I did not include paleoclimate. Well, for some reasons, but mostly because I focus it on well-established hypotheses. So there are hypotheses suggesting that some of the traits are related to present climate and present soil. So that's why I incorporated this data. I could incorporate paleo data, but yeah, perhaps it would be something interesting to look and see. But no, I didn't. I only included present data because I also think it would be difficult to work with so many data. We also could have included many other climate variables. WorldClim provides several, but it would be difficult because we are going to start to find many correlated variables and etc. So we prefer not using. We already include like three variables related to megafauna, 3 variables related to current herbivores, and it would be, I think, too much complexity, and I don't see much reason for including it right now, but it's a possibility, and I will think about it. Thank you.
Thanks. I think it could be interesting. I'm studying spines in the Atlantic forest, subtropical Atlantic forest, and I think your work would really help me to explain my data. Thanks.
Nice. I'm happy to hear that.
Okay, Marcelo, you have a question or comment?
Hello, good afternoon. It's just a comment.
Hi, Marcelo.
To see you talking, Vinicius, very nice talk. But just calling attention that many studies assume that during the last glacial maxima, we had drier climate and expansion of savannas and dry forests. And I have seen some modeling that suggests that savannas had a broader distribution in some parts of South America during the last interglacial period, before the last glacial maxima. But many studies stop in the last glacial maxima, and we do have the models ready for analysis for 130,000 years ago in the WorldClim model. So it's useful also to give a look a little before the last glacial maxima, during the interglacial period.
Interesting. I'll have a look. Thank you.
Okay, great. Pedro, you have a question?
Yes, sure. Hi, Vinicius. Thank you very much for your excellent talk. It's a great pleasure to see such a nice study. Do you think that the long-term changes in the vegetation after extinction of those animals, of these megafauna, could have involved not just the changes for individual species, but changes in closely related species, changes in whole subclades of closely related species, so that the changes are have some type of strong phylogenetic signal in the in the results that you are showing?
In what results specifically do you mean? You mean that changes in? Yeah, I'm not sure if I understand what you refer to. What changes do you think that could be influenced by phylogeny?
Well, the question is could be rephrased as whether you find a strong or a significant phylogenetic effect in the models that you are analyzing.
I think in the perspective that I chose to use, it's a bit difficult to use phylogeny because the species composition is lost once I calculate the ecoregion level mean for the traits. So we have a mean that considers many species, but I'm pretty sure that there is a phylogenetic effect on these patterns, because I often read in studies about the flora of South America that the flora of lowland South America, that includes the Cerrado, the Amazon, the Caatinga, and the Chiquitanos dry forests, is phylogenetically similar to the flora that we found in the Chaco region in southern South America, in which we have some species that are common in temperate ecosystems and some species that are more closely related to species from African savannas. So I believe that the patterns that I'm showing here do have a phylogenetic influence. Yeah.
Thank you so much.
Okay, feel free to post more questions if anyone has one. I'll ask another one. I see if other people ask some. I remember a colleague, William Bond from Cape Town, often comments that when he visited the Cerrado region, I think you were there in the talk when he mentioned this, that the Cerrado vegetation doesn't seem to be very well adapted to herbivory in terms of its flexibility, that it's very brittle, it breaks and falls apart very easily. So I'm just wondering how that observation, and obviously it's just one point, so there's not a general observation of the Cerrado biome, but how that observation fits into this pattern of seeing anti-herbivory defenses in the Cerrado biome.
Yeah, this is a very interesting question. It's something that I'm also a bit curious about. I sometimes speculate a bit about this. We have published a paper in the beginning of this year suggesting that, comparing the traits of plants in the Cerrado and the plants dominating African savannas, and we found that indeed the Cerrado plants have much weaker woods. The wood density of the Cerrado plants are very low. This is not very evident from our data because we use the ecoregion scale. So, and the Cerrado is characterized by many forest patches. So this blurs a bit the pattern, but it seems clear that Cerrado plants have low wood density, and it is clear that we have very few spinescent species. And it's curious also to see that there aren't many fossil information exactly from the Cerrado region. So we know little about the megafauna, but there are studies showing that there are plants with megafauna fruits, but they are not very numerous species, but there are some. And well, what I'm going to say now, it's an opinion, so don't take it too serious. But I have been thinking that perhaps the species from the Cerrado, the typical species from the Cerrado, used to be more restricted in distribution during the megafauna period, and then were able to expand to regions once megafauna was extinct, and then fires began to be more frequent because of the lack of control, consumer control, and then this species could become dominant. So perhaps they used it to live in mountainous areas or in areas which were refugees from megafauna or were very rare species, and then started to spread once megafauna went extinct. But it doesn't, but alternative to that, there is also the possibility that the plants in the Cerrado strongly relied on leaf traits, and we must better understand how are the leaf traits, especially chemically speaking, how are the leaf traits of the Cerrado species, and because there is also the possibility that the megafauna avoided these plants because they have very unpalatable leaves. So this is a subject that I have been interested in studying a little further, is compiling more data and also making some field work to collect data on leaf chemical traits to see how the Cerrado is different from other ecosystems in these traits.
I have a question, if I may.
Hi, Will.
Vinicius, great talk. Very interesting. And so something I've always wondered about is, are estimates of mega herbivore diversity in the Cerrado underestimated because of how acidic the soils are and the low probability that fossils will form in those conditions? And it seems like there are really, large treasure troves of fossils are really at the, on these like Lagoa Santa, where they're actually limestone karst caves, and where you have a high pH, high cation exchange capacity, and something that or a result that you presented that made me question, think about that again is when, like, for example, the relationship between pH and herbivore diversity, it could, to some extent, that be an artifact of just the conditions under which fossils can form?
Okay, well, in this case, I don't think it's an artifact because of the way the data is produced. The data here for herbivores is not based on, well, maybe a little, because of course it is indirectly based on the co-occurrence of extant species and extinct species. Well, I think I cannot answer this question because I don't know much about fossil and how it formed. Well, I know only the basics about that. So perhaps someone that a paleoecologist could better answer this question. I don't know. What I know is that in generally fossil can be more hard to form in dry places or in very cold places, but I'm also not very sure of that. And I never heard that it could be more difficult for fossil to form in acidic soils, something that perhaps, if it's correct, it may have influenced somehow.
Yeah, I think it's an issue of just under the acidity, the bones break down much more rapidly and are less likely to exist long enough to, But that, again, that's something I really don't know about.
I did notice in one of your figures, Vinicius, there seemed to be a really high biomass and abundance again in Central America, Mesoamerica, which again makes me think of these limestone soils, this karstic environment there, and I wonder whether there is a why. Otherwise, I can't understand why Mesoamerica would be mega abundant in megafauna, but similar climates in South America would have lower abundances overall. But
one thing that about the comment of Bill is that, well, we know that soil acidity generally increases with precipitation. So and we do not see, so we could expect that perhaps we have more species in areas with basic soils, and it's not what we observe when we see that the most megafauna rich region was associated with regions that were not the most the driest regions. I would say that perhaps this had a very large, a very strong effect, I would assume, but perhaps, but perhaps this what Yadvinder just said is an evidence that the fact that we have large species, well, yeah, well, I think that we generally have large species in Central America, but we do not have very rich megafauna, high richness of megafauna species. So while I see, and in fact, I did not find a relationship when I addressed the predictors of the megafauna patterns. I did not find a strong relationship with pH. Only for leaf size that I found this somewhat stronger association with pH, but for megafauna richness, it was mostly associated with cation exchange capacity and fire. First cation exchange capacity and then fire. And I found quite high variability explained by our predictors. It was much higher variability explained in the megafauna models than in the plant trait models. We found like 60% of variability explained and a large amount explained by cation exchange capacity and fire frequency, which is probably, of course, present fire frequencies do not reflect past fire frequency, but we know that they were fire-prone ecosystems and that large mammal herbivores prefer fire-prone ecosystems.
If anyone has another question, feel free to just unmute and ask it directly. I need to type it in. Yeah, I'll just follow up with one question, which is turning to the forest biome now, which is much harder. The evidence base is much harder because the preservation issues of fossils are even harder in the hot, humid, acidic soils. I've had a discussion with people like Jos Barlow and John Terborg over the years about to what extent we would have found megafauna like proboscideans within the Amazon forest. So if you could look at Africa, in Central Africa, we have the forest elephant, abundant, dispersing fruit, etc. The case in South America seems more ambiguous because the proboscideans only arrived a few million years ago with the closure of the Panama Isthmus. And so there's a question of what extent the tree species have co-evolved with proboscideans in the way that they have in Africa. And maybe that didn't provide the food supply that the proboscideans needed in the forest biome to offer elephant dispersed fruit, for example. And I don't know whether any, your analysis is really focused on the more open biomes there, but I'm wondering what insights emerge into what might have gone on in the closed forest, humid forest biome in terms of megafaunal abundance.
It's an interesting question. I think that we still knew little about the megafauna of the Amazon region, and mostly because many regions are difficult to access. I can tell much about that, only that when I was searching for diet information, I found that these proboscidid species generally had a different diet when they occurred in forests, mostly of browse, which did not occur in others. So it looks like these proboscideans were very flexible in their diet, especially the most common species, which is the Nochiomastodon platensis, was very flexible in diet. So, I would not be able to say the extent to which diet was a limitation for this species and provoked the extinction. I really don't know, but it's interesting. And I think that much more fossil information needs to be acquired before we can make strong conclusions about these forests. It recently came out a study with these paintings, painting by ancient men in the Colombia Amazon. Even in this case, it is difficult to make a very, I think, direct statement that this region was a savanna because these people walked around. So, and the region was very near, is somewhat near to the Llano savanna region. So We don't know if the actual area there was a savanna or if they walked to the lands. And we also see species that are mixed feeders. So, well, at least in the case of the Notomastodon platensis, but there were also horses. We would have to look with the tail if it is possible to identify which species were there and if those species were grazers or browsers, because this would give a general idea of whether these species occurred there because we had a savanna before or because they were actual forest species. So something to study, a lot of things to study.
Yeah, I see Felix Pim has a question that he can't use his microphone. So he's asking, given the issues we're discussing around fossils, what are your thoughts on using Sporomiella spores for determining late Pleistocene megafaunal abundances?
I'm sorry, I can't say much about that. I'm just starting with this fossil thing, so I'm generally not a paleoecologist, I'm an ecologist, so I'm sorry, I can't opine much about that.
Yeah, I don't know if anybody else wants to pitch in on that. I guess part of the challenge is finding good depositional environments where you can get a good, nice time coded, like a lake or something, where you can get nice time sequences. I know Mark Bush and others have looked for these around the Amazon, and they tend to have a lot in the Western Amazon and the Andes where there are lakes. But once you get into the mass of the Central Amazon, at least, that becomes quite difficult to find good locations.
Yeah.
Okay, so if anybody's got a final question, feel free to just unmute. If not, I'd like to thank Vinicius. I encourage you to switch on your mics and your cameras so he can have a real audience. Give him a round of applause as well. But thank you for absolutely fascinating.
Thank you. Thank you very much. Okay,
so as I mentioned, we'll pop this onto our YouTube channel. So if you just look at my YouTube channel, you'll find this seminar and all the previous ones as well. So so you could look there. But congratulations, Vinicius. Absolutely fascinating. This is a question that I know several of us have been thinking about for a while, and it's really nice to see a nice new data set and evidence base emerging to answer some questions, but also open up many more questions, and it's inevitably new data always does. So congratulations on your work.
Thank you. It was a great pleasure.
And thank you, everyone. Have a nice winter break, and hopefully see many of you in 2021. Okay. Bye-bye.
Bye.
Bye. Bye-bye.
Bye. Bye.